Hello, my name is Willette Streets. This is Bio 225. Today's date is 9/29, 2018. And today I'm going to be doing the motility testing. This is week 4. I got a little mixed up on the lab part. So I'm going to explain to you what I did, and then I'll show you the results. So the first thing was with the motility testing, we had to straighten 2 paper clips and um, and put the 2 paper clips in alcohol. So these are the 2 paper clips that I straightened out, and I have them sitting here in alcohol. And then we had to use a permanent marker to label each motility tube. So one would be labeled for E. coli, and then the other one would be labeled for Staphylococcus epidermis. Okay, and then the next thing that we hold on, my phone is ringing. So then the next thing that we had to do, we would light our candle, and I kind of already did it. So we would light our candle, and the candle is for sterilization. So and then we would remove the lid from the motility testing tube, and I had already did that. And what we would do once we remove it, we would move it and sterilize it with the heat from the candle. And then after we did that, we would take a paper clip and shake it to dry off, and then we would remove from our culture here, and we would take the paper clip and we would go through the culture and get some of the colonies. And so then after that, after we stab the paper clip into the agar, then the next thing we would do, we would flame the opening of the motility agar again. We would flame it again, and hold on, my phone is ringing. So sorry about that. So we would flame it again for sterilization, and then we would close it up. And so I did the exact same procedure for the epidermis. I did the exact same procedure. Here's my specimen here that I was able to stab for the next procedure, and this is the next one here. And so after we did that, it had to incubate for 48 hours. And then then we had to observe it for motility. I'm sorry. We had to observe, I'm sorry. It had to incubate for 48 hours for microbial growth. If no growth is observed, and then we had to incubate it for another 24 hours. Well, after 48 hours, I did have some growth to it. So for the first one, you can see for the E. coli, you can see some growth inside there. And then for my next one, which was the S. epidermis, you can see some growth. Kind of looks really cool there too. You can see some growth and some specimens, and that was after 48 hours. And so after we observe each tube, noting the turbidity of the agar and the definition of the original stab line, a clearly defined stab line in a clear agar is a negative result. And so after I take my pictures, then I will record it on my data table 2. I will record it. And me show you. Let's see. You can see here. Here's my my E. coli dish, my epi dish. Here's all my little stuff here. And like I said, I kind of got mixed up on it. And so here's everything here. And again, my name is Willette Streets. This is Bio 225. Today's date is 9/29/2018. Thank you.